Good day and welcome to another very entertaining video. I'd like to start off our bone unit today by talking about hyaline cartilage. I've got a structure here, the knee joint that we're looking at. You can see the patella, you can see the femur on the top, the tibia on the bottom. But what I'd really like to focus on is what's in blue, what we call articular cartilage and also the meniscus. These are coverings at the end of the bone that provide smooth, lubricative surface for this joint to function properly. And this is hyaline cartilage. When we think of hyaline cartilage, we're thinking tough, pliable, sometimes gelatinous, lubricative. These all describe attributes of hyaline cartilage. And many times, knee problems can be associated specifically with disruption of this cartilage, where you wear down these surfaces over years of use and abuse, and pretty soon you got bone grinding on bone. And while that's cool, I think what's even more important for us to understand is the fact that at one time, all of the bone that you see here, the patella, the distal femur, the tibia, all of it, this used to be hyaline cartilage. And so from our point of view, we want to understand hyaline cartilage not necessarily for what it does for us today, which is important, but also for what it did for us embryonically, which was to provide a scaffolding for endochondral ossification, the formation of bone on top of cartilage. So with that direction, let's take a look at what hyaline cartilage is. In its most simplistic form, hyaline cartilage is simply two things: fibers plus what we call ground substance. The cell responsible for the production of these fibers and ground substance is a cell called a chondroblast. And as we've been trying to link cartilage and bone together, I think it's important to note that the chondroblast has as its progenitor cell a cell called the osteochondral progenitor cell. And it's the same ancestral cell type that actually gives rise to the osteoblast, which is the cell involved in bone formation. And so we see that the osteoblast and the chondroblast, they're actually cousins. And so a lot of the patterns that we'll see in cartilage formation will be applicable also to bone formation. Now, we will see a bit of a journey as this chondroblast turns into, I don't think differentiation is the right word because that's not quite what happens, but it can become what's called a chondrocyte. What's the difference between a chondroblast and a chondrocyte? Simply location. The chondroblast has not yet surrounded itself by its extracellular matrix, whereas the chondrocyte is surrounded by extracellular matrix. And because it's walled in, it essentially lives in its own little hole, its own little cave. And we call this cave a little lacuna. And we'll show you what that means in just a second. But for right now, what we want to do is we want to focus on this extracellular matrix, right? Because it's the formation of that matrix that makes these cells unique. And we already said it consists of fibers and ground substance. So first, let's look at the fibers. The dominant fibers that we're interested in are collagen, of course, and to some extent elastin. I don't think we need to detail elastin too much, just recognize it is what it is, right? It's a very elastic structural protein that provides stretchability, whereas collagen has high tensile strength, it's a super abundant protein, and we want to take a look at that one. So let's consider collagen for just a second. We said it has high tensile strength, which simply means it resists being stretched too much. I can pull on this a lot, and it's strong. It is the most abundant protein in mammals, simply because we find it in things like tendons, ligaments, also in bone, of course cartilage, which is what we're talking about, and also skin. The final formation of the protein and what gives it its high tensile strength is the fact that it exists in a triple helix. Might look something like this. All wound together, the combination of these structural proteins give collagen its very high tensile strength. Okay, the other part of this extracellular matrix that we discussed was called ground substance, so let's take a look at that. The easiest way to think about ground substance is one word: proteoglycan. Of course, maybe you're not familiar with that word, but we can easily break this down into two different words: proteo and glycan. So what is this? This is simply protein plus sugar. right? And by sugar, what are we really talking about? We're talking about carbohydrates. Now, why is this important? Well, the protein provides a basic scaffolding for which to attach the sugar. The player here is really the carbohydrates. Why is ground substance so reliant on carbohydrates? Well, remember, these sugars, because they're so polar, are very hydrophilic. The carbohydrates simply act as nature's molecular sponge. And if I'm looking for cushioning or lubrication or a nice, smooth, easy surface to glide on, like articular cartilage, I want water there. And so the sugar pulls in the water and holds on to it because of its solubility. Now, we're not talking about monosaccharides or disaccharides. We're talking about very large polysaccharide chains. Maybe as an example, let's talk about one of these proteoglycans, a common one in the body, hyaluronic acid. This is one of the longest glycosaminoglycans, thousands of monosaccharides, and it is the major component of the extracellular matrix. It's stiff, it's gelatinous, it's lubricative, and it's repeating dimers of two specific monosaccharides, N-acetylglucosamine and glucuronic acid. If you're interested in what this looks like, we could draw it out real fast. There's the glucuronic acid, and there's the N-acetylglucosamine, another monomer, slight different modifications, held together by a beta-1,3 glycosidic linkage. And we're going to take this dimer and we're going to repeat it thousands of times to build hyaluronic acid. So let's add another dimer on there. This is a beta-1,4 linkage. And we can see how this is going to build. That's when we talk about hyaluronic acid, when we're talking about glycosaminoglycans, proteoglycan, this is the idea. It's a bunch of sugars. Now, the protein part, we can further elaborate on that. So let's zoom out for just a second. And if we were to continue this hyaluronic acid polysaccharide, we would see at regular repeating intervals these little protein links along the hyaluronic acid, to which we would add a core protein sticking out. In this case, the protein would be a grechin, and the whole purpose of that core protein is simply to add more sugars. So let's add some chondroitin sulfate. These are going to be dimers between glucanac and glucuronic acid. And let's also add some keratin sulfate. Again, another polysaccharide, this one made with galactose and glucanac. The key point again being, I'm not so much interested in the types of sugars that are in there, but simply the understanding that the ground substance is designed specifically to provide a molecular sponge. Lots of ways to bring in water. That's what the carbohydrates are doing, and we're strengthening it with the core proteins. That's the idea of proteoglycan, protein and sugar, where protein provides a little bit of structure and sugar attracts the water. Okay, so now that we have a basic understanding of what hyaline cartilage is all about, let's actually look at cartilage itself, the structure. If I'm looking at a histology slide, what would I expect to see if I'm looking for hyaline cartilage? Well, to do that, let's first start with the outer layer boundaries. Let's start with the perichondrium. Interesting that the perichondrium is absent in articular cartilage, like we were showing on the knee, but it will be present in the hyaline cartilage that we would see in the airways surrounding the bronchioles, for example. And this is going to consist of a fibrous layer and a chondrogenic layer. In the fibrous layer, we have a bunch of fibroblasts. And the fibroblasts are just my primary source throughout the body of collagen-producing cells. And so that's what these cells are doing. They are producing collagen. So let's throw some collagen down here. Beneath those cells, now into the chondrogenic layer, this is where we find our chondroblasts. Now remember, the difference between the chondroblast and the chondrocyte, these haven't surrounded themselves with all their extracellular matrix. So what does that look like? Well, now getting into the actual hyaline cartilage, we would expect to see these lacunae that we mentioned. These are going to be open holes, cavities really, and the chondrocytes are going to be the cells inside. Give them some nuclei. Not that the others don't have any, they certainly do, but this helps us see what's going on. So the space itself is called a lacuna, plural lacunae, and the cell inside is now a chondrocyte. And remember, they've surrounded themselves with their extracellular matrix, fibers plus ground substance. Throw in some fibers, some ground substance. That's how we're going to recognize hyaline cartilage. Now, there's a couple of things worth noting. Number one, cartilage is avascular. This is why it takes so long to repair. It doesn't have a really good source of nutrients. The outer perichondrium is vascular, but that's where it stops. Down here in the cartilage itself, no blood. The last thing we want to talk about simply is types of growth. There's two types we want to mention. The first one is appositional. The appositional involves the chondroblasts. If we draw a chondrogenic layer here, what I want you to appreciate is that as we add layers, these chondroblasts will contribute to the growth of layers. And so here the growth, appositional growth, is all about increased width as I'm adding layers. This is in contrast to the second type of growth we want to talk about, called interstitial growth. Interstitial is all about the chondrocytes. And hopefully you can appreciate this is going to be a little bit more multidimensional. As we draw our lacunae, we continue to push outward, but really this happens in all directions. Oftentimes people will say appositional is width and interstitial is length. And while this certainly contributes to length, I want you to appreciate that this is simply growth from within, whereas appositional is adding layers on the outside. That's the difference. So in summary, if we back up, hyaline cartilage is an important part of joints in the body. It also plays a role in supporting the airways. But what we're really interested in is understanding that nearly all of the bones in your body first started as hyaline cartilage. And so we want to understand how to build it. The hyaline cartilage itself is made up of fibers plus ground substance and the cells that make them. These are either chondroblasts or, if they're surrounded by their ECM, chondrocytes. As we separate fibers from ground substance, the dominant fiber is collagen, very high in tensile strength, and exists in this triple helix formation, the most abundant protein in the body. The ground substance is simply proteoglycan, right? Protein and carbohydrate. And the importance of carbohydrate is that it acts as a molecular sponge to draw in water, to provide the cushioning and the lubrication and everything that hyaline cartilage is designed to do. If we actually look at the structure, we can see in some instances a perichondrium where the fibroblasts are on the outside. This is a vascular portion. And then we get into the chondroblast layer, and now we're avascular. The chondroblasts are on the outside. They haven't yet surrounded themselves by all of their extracellular matrix. And when they do, we can see in the deeper layers these lacunae, these classic little caves where these cells are living, the chondrocytes. As we add layer upon layer from the chondroblasts, this is appositional growth, whereas interstitial growth occurs from the chondrocytes themselves, pushing outward and expanding from within.